May I audible?
Yeah.
And are you able to see my slides?
Yes.
Yeah. So thank you so much to the organizers, Dr. Geriant, Dr. Poets Hansen, Dr. Van Larry, for giving me an opportunity to discuss about the story that is behind to banana, where we are trying to improve the nutrition quality by using genetic engineering approaches, both genome editing as well as transgenic. So thank you so much to the organizers. So before going to details, we have seen and the Victoria presentation where she has faced so many problems regarding trials of genome-edited rice. But fortunately, in India, that government is very supporting. In March 2022, they have exempted that genome-edited crop plants. Those are considered under the SDN1 or SDN2. So in this case, if we are proof that there is no exogenous gene introduced in the genome of crop plants, it will not be followed under the biosafety assessment as we are following for transgenic. So this is the really great advancement in Indian scientific community, specifically those are working on biotechnological approaches in plants. So before going to details about my presentation, that is mostly scientific presentation, I would like to introduce briefly about my organization. It's a National Agri-Food Biotechnology Institute. In short, it's known as a NABI. The institute started in 2010 in Mohali, that is located in the state of Punjab, just near to New Delhi. So my institute vision is very clear to provide food and nutritional security to all through agri-food biotechnology research and innovation. By following the vision of my institute, I started working on biofortification. Biofortification means to enhance the nutrition quality of crop plants. And I am utilizing biotechnological approaches. That is basically a gain of function or overexpression. In this case, I have used transgenics or cisgenic approach. The second one is the loss of function, where we would like to reduce the anti-nutrient factors, as well as we are trying to modify the metabolic pathway to enhance the certain nutrition components in crop plants. And in this case, we are preferring to use genome editing approach that is based on the CRISPR-Cas. So as I mentioned, we are working on the nutritional problem, and you know that hidden hunger or micronutrient deficiency is the major problem. That is basically a global problem. And it is reported that more than 2 billion people are suffering from major micronutrient deficiency. That is vitamin A, C, iron, zinc, iodine, and folate. And micronutrients basically a small fraction of the balanced diet and play major roles in human metabolic processes. So if you are not getting sufficient micronutrients in your diet, you will considered under the category of hidden hunger or micronutrient deficient, and it is responsible for many diseases and even for mortality. So these micronutrients you can get from diverse dietary sources. Like if you are eating animal foods or diverse foods, then you are able to get these micronutrients. But it is not feasible for most of the pupils. Those are living in the underdeveloped countries or developing countries, because those pupils are mostly dependent on the staple crop plants. So crop basically or plants are the major source of micronutrients. If those plants are deficient with the nutrients like iron, zinc, or ProvitaminA, then subsequently it will lead to the malnutrition problem or micronutrient deficiency in the population. And this micronutrient deficiency you can be resolved by three different categories. One is the nutrient supplementation, dietary modifications, and biofortifications. So biofortification is one of the very promising approach because it's a one-time investment, and it is categorized in the three categories. One is the traditional breeding, second one is the agronomic approach, and third one is the genetic engineering. So we all know about the traditional breeding limitations. It is only that the characters are present in the crop plants, related crop plants, then only be introduced by breeding approach. Agronomic approach is basically not a feasible approach, or it's required a investment again and again. But genetic engineering is basically a one-time investment. If you are modifying the genome of a crop plants, either by genetic engineering like transgenics or genome editing, it will be stable for generation to generation, and you will get that characteristics. So this is the advantage of genetic engineering. So here you can see that genome editing has certain advantages. We know that it can be implemented for improving various characteristics, like we can improve the biotic abiotic stress tolerance, we can enhance the micronutrient contents, we can develop the seedless crops and also reduce the anti-nutrients factors. But this approach also needs some kind of limitations. Like first and most important is that which crop you would like to modify by genome editing, the sequence of genome should be known. Otherwise, you will get off-target effects or will not get a desirable results after editing. And second one is that you have to be optimize the protocol of tissue culture in a particular crop plants or a variety of plants that you would like to improve. The third one or fourth one is basically a delivery mechanism. Which kind of delivery you would like to implement in your system, like particle bombardment, agrobacterial transformations, or protoplast-based editing. So as I am working on clonally propagated crops like banana, and similarly in other crops like potato, sugarcane, citrus, and cassava have certain limitations as well as advantages if you are working on genome editing. If you will see the merits or advantages, it will facilitate the editing of multiple alleles simultaneously. Specifically, these are the polyploid crops. You all the alleles you can be simultaneously added by using genome editing. And it will also work like a self-containment because the crossing or breeding is not possible. So gene will not flow to the related germplasm or varieties. This is another advantage. But demerit is that if that Cas9 you are using is going to be integrated in the genome, it will be not segregated out. So it will be remain in the genome, and that why it will be considered like transgenics. Most of these staple crops or like clonally propagated crops are considered recalcitrant for tissue culture and genetic transformation. So you have to be optimize the protocol for these crops. It's very much required. The solution of these kind of problems is basically you can use a protoplast based transient expression of genome editing regions. Then you can also be used like plant growth promoting factors. Those have report already been published in different journals. So people are trying to resolve this transgenic issues where we can express transiently the regions of genome editing, and it will not going to be integrated in the genome. And in this case, protoplast is one of the best solutions. So in my case, I have used banana, and banana has so many advantages. If you will see, it's a tropical food crops cultivated in more than 130 countries, and it is the second largest food produce just after the citrus. It contributes nearly 16% of the total world food production. In case of India, India is the largest producer of banana and contributing nearly 26% of the world food productions. The fruit is available throughout the year at the reasonable price and most suitable and choicest foods for all age group people like infants, old as well as young people can easily eat this fruit. But why we have to work on that genetic engineering approach or biotechnological approaches? That because as I already told, the all cultivated varieties of banana having triploid genome. So those are genetically sterile. So it is not possible to improve those banana varieties by using breeding approach. The only solution to enhance the nutrition value or desirable traits in banana by using genetic engineering. So in case of I am working to enhance the ProvitaminA, that is the precursor of vitamin A. Why we have chosen this particular traits? Because if you will see the global status of that vitamin A deficiency, it's a very severe. It is reported that more than 6.5 lakh children die per year because they are not getting vitamin A sufficiently in their diet. And another more than 3.5 lakh children go permanently blind just because of deficiency of vitamin A in their diet. Vitamin A deficiency is also responsible for mortality, blindness, night blindness, impaired immune system development, as well as impaired brain development. In case of India, you can see that around 57% of children, those have those are having less than 5 years age are deficient with vitamin A. And it is also reported that more than 3 lakh children die per year because they are not getting sufficient vitamin A in their diet. This is really a serious problem, specifically in India and many other Southeast Asian countries and African countries. So how you will get vitamin A? There are different sources of vitamin A, like you can get from animal sources or plant sources. But in case of animal, it is in the form of retinol. But in plant, it is in the pigmented carotenoids known as a provitamin A. Alpha carotene, beta carotene, gamma carotene are the basically carotenoids. Those can be converted into the vitamin A in our body. And it is reported that approximately 375 billion people worldwide and around 30% of the Indian population is vegetarian. So that's why we are thinking to use a plant as the major source for the enhancement of beta carotene or provitamin A. Then question is that how much vitamin A we have to be enhanced in banana. For this, we have taken the information from Indian Council of Medical Research, and it is clearly showing that men and women will get 1000 microgram vitamin A per day to get sufficient requirements of this particular nutrition. And the conversion of beta carotene to vitamin is 1:6. So that if you would like to get 1000 microgram vitamin A in your diet, you have to eat 6000 microgram beta carotene. So based on that, we have screened the Indian banana germplasm and noted that one of the most common cultivated variety, Grand 9, contained only 400 to 500 microgram beta carotene in 100 gram of their eatable portion, this pulp. So if we would like to get 100% RDA, that is at 6000 microgram, we have to be increase nearly 15-fold content of beta carotene in the single banana. So this is that basically calculations, and based on that, we started working on banana to enhance this particular nutrition. This is the well-known carotenoids pathway in plants, where you can see that in the through MEP pathway, this one particular gene, deoxyxylose 5-phosphate synthase, DXase, provides substrate flux in the carotenoids pathway, and subsequently geranyl diphosphate converted into the phytine by the activity of phytine synthase, and this phytine is subsequently converted into the lycopene by several enzymatic steps. Lycopene is diverted into the two directions. One is the alpha carotene or lutein biosynthesis with the activity of lycopene epsilon cyclase, while lycopene beta cyclase is responsible for the conversion of lycopene to the beta carotene. This beta carotene is going to be degraded by the other enzymatic steps like BCH and CCDs responsible for the beta carotene degradation. So based on this pathway, I started to make my hypothesis or approach how we can enhance the beta carotene. So I followed two approaches. One is the gain of function, that is basically overexpression of these two genes. Those are available in the upper pathways of beta carotene, DXase and phytine synthase. And for CRISPR approach, I have used other genes like lycopene epsilon cyclase that is responsible for the diversion of lycopene into the other pathway sites. So if we edit this gene, the possibility is that flux can go to synthesize beta carotene. Similarly, that we have targeted BCH and CCDs genes, those are basically responsible for the degradation of beta carotene. So if we edit or silence these genes by using CRISPR, the beta carotene will be stable. So, and PDS we have used as a proof of concept to just standardize and prove that genome editing is possible in banana, that I will let you know in my subsequent slides. So first, as I mentioned, it's the most important to optimize the protocol of regeneration and transformation. So you can see we have very efficient protocol of regeneration where we have used embryogenic callus, and this embryogenic callus we have used to develop cell suspension culture. It's a liquid media, and here you can see thousands or millions of embryonic cells. Those are in the liquid media that we can maintain up to the 2 years by routine subculturing. And when we transfer this embryogenic cells in the semi-solid media, it is going to be regenerated. So this protocol is very efficient and well established in my lab. And from 1 ml of cell suspension, we are able to generate 1000 of banana plants. Based on this efficiency of tissue culture, we started working on genetic transformation. And for that, I have made this pipelines or steps where first the gene that we are going to be utilized for genetic engineering, we have to be first used for in silico analysis. So we have to be characterize that gene that is really responsible for a particular traits. Then after characterization, the bacterial complementation system, and functionality of that particular gene, we started that genetic transformation by transgenics or genome editing approach. As I mentioned, we have very efficient protocol of embryogenic cell suspension culture. After transformation of this embryogenic suspension culture, we have to regenerate by using our optimized protocol. And by following the guidelines of Government of India for biosafety regulations, we grow those plants in greenhouses initially, and subsequently those plants transfer into the big net houses because banana is a tall herbaceous plant. So all plants has to be grown in the big net houses. And when fruiting is started, we have to perform HPLC analysis to make sure that our particular traits of beta carotene has been enhanced or not. So for this, we have published several research articles. If somebody is really interested to go in details, they can read these articles. So here you can see initially we have generated more than 500 banana events. Those are some of them are transgenics and most of them are genome edited lines. Then after that, we had transferred those plants into big net houses, you can see, by following the biosafety guidelines of Government of India. And then fruit has been come out and we started analysis of these plants. So now I will just briefly introduce about my genome editing work. Here I already mentioned we have started to work on PDS, that is a 13-decentrase gene that is responsible for the carotenoids biosynthesis as well as that chlorophyll biosynthesis. If you will edit this gene, you will get albino phenotypes and it will prove that your approach is workable. And the second target is lycopene epsilon cyclase. This we edited it by using genome editing in case of banana. Then third target we have proved by publishing articles in Journal of Experimental Botany in last year, where we edited this CCD gene and we have shown the increased content of beta carotene. So these work have already been published in various journals. And recently we have also published some interesting review articles. If somebody is interested, they can go and read these review articles published in 2020, 22, 2023 and 24 recently. So first target is PDS. As I mentioned, it has been used as a proof of concept, and this PDS gene is responsible for the beta carotene biosynthesis. That is basically a ProvitaminA, and also responsible for chlorophyll biosynthesis. If you will edit or silence this gene, you will get albino phenotypes, and that's why we have used as a proof of concept in banana, and we have generated number of embryos by editing of PDS gene in banana genome. You can see in the tissue culture stage, it has been shown albino phenotypes as compared to the control embryos. Some of the plants have been grown and they have completely turned into the albino, and we have sequenced those edited lines. We have seen that stop codon has been generated due to the frameshift mutations or editing, deletion or addition of some nucleotides. And based on that, we have estimated the chlorophyll content as well as total carotenoids of these edited lines and find out that as compared to the control, the content of chlorophyll and carotenoids has been significantly decreased. So based on that work we have published in Journal Functional and Integrative Genomics in 2018, and that was the first report in the world that had been shown that genome editing is possible in case of banana. Then my second target is lycopene epsilon cyclase. As I mentioned, this particular lycopene epsilon cyclase is responsible for the diversion of lycopene resources into the lutein or alpha carotene biosynthesis. But our target molecules is beta carotene. So that's why we have designed the CRISPR construct that have been used the fifth number of exon to design this guide RNA. And subsequently, we had performed transformation. And then we have generated number of lines. Here, after generation of lines, we have performed the sequencing. And you can see the catalogs of that chromatogram has been generated, and where we have seen the addition and deletion of certain nucleotides. We have made sure that there is editing. Then we have performed T7 endonuclease assays of these edited lines. And when we estimated the carotenoids content in the edited fruits of banana, we have find out the content of that lutein and alpha protein has been decreased as compared to control, while the content of beta carotene has been significantly increased in some of the edited lines. And due to the high carotenoid depositions, you can see the edited lines has been turned into the dark yellow color as compared to the control. So when we estimated that carotenoids content in edited lines, we noted that nearly 6-fold high content of beta carotene increased in some of the lines. We have also performed some agromorphological analysis in our net house grown plants, and we noted that as compared to the control, the GN9 lines, those are showing heterozygous editing that has been proved by T7 endonuclease is comparable to the control while showing nearly six-fold high content of beta carotene. So that work we had published in Metabolic Engineering 2020, and that was the first report in banana to improve any nutrition trait by using precise genometric approach. Now my second target is that basically a CCD gene that is responsible for the beta carotene degradation. So for this, we have identified two contrasting cultivars of banana, Rasthali and Nentran. Those we are growing at our research field. And when we had performed that expression analysis in the fruit developmental stage of these two contrasting cultivars, we find out CCD4, CCD1, and CCD8A are showing abundance amount of expression in the fruit pulp. So based on that, we have taken the different food developmental stages of these two contrasting cultivar, Rasthali and Nenden. This Rasthali variety is known for low beta carotene content, while Nenden variety is known for high beta carotene content. So we have noted that this in case of Rasthali, the CCD4 is showing very high expression at the early food developmental stages. While the other variety Nenden, those are known for high beta carotene depositions in fruits, it is not showing any kind of CCD's expression at the high amount in different developmental stages of fruits. So based on that, we concluded that CCD4 might be the potential target for beta carotene degradation in case of Rasthali cultivar. This cultivar is most commonly grown in several parts of India. So we have designed the strategy to prove that this CCD from Rasthali is really responsible for the beta carotene degradation. We had cloned this CCD4 from Rasthali and performed complementation study in bacteria that shows that degradation of beta carotene. And subsequently, we also performed that overexpression in Arabidopsis to make sure that after overexpression, what is the impact of that carotenoids content. And we noted that in the seeds of overexpressing lines of Arabidopsis, the content has been degraded. And then also we have performed enzymatic assays where we have that purified that enzymes of CCD4 from Rasthali and performed GCMS analysis by using different substrates of carotenoids pathway, and we find out that CCD4 is responsible for the degradation of beta carotene 10 apobeta carotenol by the formation of and based on that, the CCD4 we had considered for designing the constructs, and that has been performed in the protoplast of banana that we have optimized in our laboratory, and we have noted down the editing of CCD4. Then we also used that particle bombardment approach by using the construct. There is no left-right border present. So subsequently transformation and regeneration, there is no selection marker. We have generated nearly 300 plants, and we had performed sequencing and noted down two plants are showing that editing, and there is no integration of Cas protein in the genome. Based on that, these lines have been considered as a non-transgenic. And then we had performed the analysis in the root as well as in the leaf tissues of these two edited lines, and it shows nearly 2 to 3-fold high content of beta carotene depositions as compared to the control. We also perform expression analysis of different genes, and that work was published in Journal of Experimental Botany in 2020-22. The foods has been recently collected from the field of these edited two lines, and now we are going to perform their carotenoids estimation to make sure how much that carotenoids has been increased in this non-transgenic edited line. This is the protocol of that we have recently optimized, that is based on the protoplast. You can see we have used the vector GFB based, and it is showing very high expression in the protoplast, and this protoplast we have optimized by using the regeneration methods, and it is showing regeneration, although the efficiency is very less. And this optimized protocol and conditions for protoplast regeneration, we are trying to implement the multiple targeting of all three genes of carotenoids pathway, that is a lycopene epsilon cyclase, PCH, and CCD. The construct has already been designed, and now we are trying to implement these constructs in the protoplast transfections. So we have to be generate non-transgenic line and with the high carotenoids content by using genome editing. My second work is related to overexpression. As I mentioned, DXSS provides substrate flux in the carotenoids pathway. Again, these two contrasting cultivar Rastali and Linden we have considered for the expression of DXS genes, and we find out that DXS2 gene is showing that promising roles while overexpression in that complementation study, and it has been shown the high depositions of beta carotene. Based on that, DXS2 from Linden cultivar we had considered for the designing of construct. We had generated the plants. You can see in the net house, those plants have been generated the fruits, and it is comparable to control. There is no changes in that phenotypes. When we cut that fruits, you can see that content of beta carotene has been increased nearly 20 to 30 fold high as compared to the control, and it is clearly reflecting by the high that dark yellow color or golden color. That's why it's known as a golden banana that we have developed by this study. Then subsequently, we had performed the Caco cell line based bioavailability to make sure that our increased carotenoids beta carotene is basically a bioavailable or not. In this case, we have noted down that single banana is sufficient to provide 100% recommended dietary allowance, that is 1000 microgram of vitamin A. So my work is basically now this biofortified banana is in the second phase where we are performing multi-location event selection trials in open field in different places of India. We have considered 5 locations, and also we are taking help from that one of the biosafety experts, BCI agency in India. Those are supporting us to follow all that regulatory guidelines or biosafety guidelines in India. We got that all regulatory public permissions from Government of India, like from RCGM, GEAC, to perform this event selection trials in open condition. So in case of this, you can see this is the map of India. We have considered 4 different geographical locations: south, north, east, and west. And these are the different organizations or institutes where we had started that open event selection trials of our biofortified banana. This is the place, Mohali, where I am working, and the plants has been soon started that fruiting. But other locations like Assam as well as Gujarat, fruiting has already been started, and that our experts committee have recently visited over there, and they are very satisfied and happy to see the regulation that we are following for our open trials, as well as other trials is National Research Center of Banana, that is in Krishnapalli in Tamil Nadu. They have also started collecting the fruits, and these fruits and agronomical analysis will let us know which variety is really most suitable or which lines is most suitable to go ahead for BRL or advanced trials in the open conditions. So this is the journey of golden banana from lab to land. That work was started in 2013-14, where we have optimized the protocol of regeneration. Then we have designed the constructs. We had performed transformation. Then we had generated the events and plants and grown in the net house, because banana takes nearly 2 years time to reach in the fruiting stage. After collection of fruit, we have seen that some of the lines have been shown very encouraging results, and those lines we have considered for multiplication. And now that we are performing multi-location event selection trials to see the agronomical as well as that nutritional performance in different locations in India. This is the news published in that articles. That is one of the famous newspaper in India, Times of India. In last year, it has been clearly mentioned after mustard, genetically banana, rubber, and maize on the way. So Government of India is very positive. They are very supporting this technology, and we are hoping that soon this banana will be in the field for consumptions. Also, we are trying to implement knock-in approach for this DXS gene near to ACO locus. That is basically a ethylene responsible gene that is already identified in the banana genome. We would like to develop that basically genome edited banana by overexpressing DXS by a precise editing. So in summary, that this is a layman language slides that is clearly depicting that we can clearly mention that this is the requirements of vitamin A, that is 1000 microgram per day for Indian populations. And beta carotene is required 6000 microgram per day to get 100% recommended dietary allowance for vitamin A. And this is basically ordinary banana. It is saying if you would like to get me that nutritional requirements of vitamin A, you have to eat me in more numbers. But our golden banana, that is a super banana, is saying I am single sufficient to provide 100% RDA. So this is just a layman language so that people normal people can also be understand the advantages of this technology. So this is that one of the interview was conducted in 2021. And if somebody is really interested to see the story behind to develop this golden banana in laboratory to the field condition, they can see this particular interview to get more information about this. I am really thankful to that my colleagues, my basically lab members, those have some of them have already been well settled in different places in India, as well as some of them got permanent positions in India in different organizations. And this is the current lab members, those are actively working in this particular project as well as in other activities in the lab. So I am really thankful to have these kind of colleagues, those are really responsible to reach at this stage. So with this, I would like to thankfully acknowledge to our collaborators in India as well as in abroad, my funding agency, BIRAC, as well as Department of Biotechnology, our directors who are supporting me for this technology. So with this, I would like to end my presentation. Thank you so much.
Thank you, Siddharth, for this nice work and presenting us. And I also see congratulations for your excellent work already in the chat. So to open the discussion, I see already some questions in the chat typed. So the first one is, how many edited events were obtained and how was the trait-specific variation among them?
Initially, when we have generated lycopene's epsilon cyclase editing, we have generated 8 events. And out of 8, we find out that editing in the 6 events. And out of 6 events, we find one of most promising events that is showing 6-fold high content of beta carotene. So this event we have multiplied in the laboratory and we are going for next trials. Although one of the limitation of this lycopene epsilon cyclase edited line is that it's a basically considered transgenic because there is a Cas9 integrated in the genome. That is a vector-based approach. But subsequently, when we edited CCD4 gene, it's a non-transgenic approach. In this case, we have 2 edited lines and we are performing that trials in that net house conditions.
And is it correct, so that we understood it well, that out of 300 plants, 2 showed Cas9 editing and they both were transgene-free?
Yeah, actually what happened, we have used that vector. There is no left-right border present and there is no hygromycin or kanamycin selection gene. So we have to generate plants without the selection present. There is no selection pressure. So based on that, we have generated nearly 300 and 400 plants. And there is no option, only we have to go for sequencing. So all plants we have gone for sequencing. And then we had find out that only 2 plants are showing editing. And some of these 2 plants are basically replicates. You can see that same plants has been multiplied again and again in tissue culture stage. So those are the replicates of the same events. So basically we can say only 2 events originally have been shown editing.
And there was no regeneration of the transgenic plants?
In this case, by using vector backbone? No,
yes,
we didn't find out any transgenic. Because only 2 events have been shown, those have been edited, as well as there is no Cas9 protein integrated.
If they are the good ones. How was the keeping quality of modified banana? Was there any penalty on yield or any other nutrients or micronutrients?
This is a very good and important question. That's why we are performing multi-location event selection trials. As I mentioned that we are performing these trials in different locations of India. So we have to see that real performance after getting that yield of these plants. Those are showing that high content of beta carotene, as well as we have to just perform proximate analysis by collecting fruits of these lines. So only based on that we can say what is happening. But in case of initially when we had performed that analysis in our net house plants, those are comparable to control. We didn't notice any kind of yield penalty or negative impact when we perform our experiments in the net house condition. But now that I have shown that plants are growing in the open field condition in different geographical locations in India, so we have to be analyze those plants and let update after some time.
And do you need to go to clinical trials for instance, the golden banana?
Not clinical, but we will perform some study in the mice. So we have to make sure that it is our beta carotene is converting into the vitamin A. And if government will allow, then we will definitely go for human trials.
Then another question is about what is the frequency of DNA-free edited plants using protoplasts?
It's very less.
It's less.
Yeah. Basically, we can say that protoplast transfection is easy, but regeneration of edited lines is challenging. So now we have optimized the protocol of protoplast regeneration, but efficiency is very low. We are refining this protocol, and soon we are hoping that it will be an efficient protocol with us.
So this maybe answers then the next question. Which step was the most challenging, or is there another step that is even more challenging?
Only that regeneration of protoplast, we can say. We are still struggling for that.
And then I see a last question for the moment in the chat. Are there any issues with eating too much beta-carotene or vitamin A? Do you have to take this into consideration?
This is really very important and interesting questions. I have usually many people ask me these questions. What happened then when we will eat number of bananas? Those are basically rich with beta carotene. But good thing is that beta carotene is not only responsible to convert into the vitamin A. Beta carotene is a very strong antioxidant. It has so many basically metabolic processes in our body. It is responsible for many activities. So if it is excess in the amount in our body, it will be utilized for other purposes. But this will not happen in the case of vitamin A. If you are getting vitamin A directly in the form of supplementations or in tablets, it is going to be deposited in your liver and it will damage the liver and might be responsible for other diseases. But beta carotene will not create any harm. I have seen that literature and reports, and it is clearly mentioning beta carotene will not harm to the human if you will get even in excess amount.
So see that this is the last question that was in the chat. So if no more